evening, everybody.
All right.
Hi. Hi.
All right, I'm just taking attendance real quick, and then we're going to dive in. I know the syllabus says that we would have an explain it to the media quiz today, but y'all just took an exam, so I'm going to move that to Monday. I'm sure no one's like super sad about not doing a quiz today. So if you are, I apologize. But basically, just wanted to give you some breathing time and also acknowledge that sometimes right after exams, you don't have time to do the next set of reading like you want to. So Monday will be the next Explain It to the Media quiz at the start of class. So once I get everybody checked in here, then what we will be doing is starting talking about sensation and perception. So this is a bit of a longer lecture like the biopsych one was. We'll talk about all the senses and how the brain interacts with them. So it's a fun section to cover because there is so much cool information about what people can do and what people see and how people see it. And I know one of the things someone asked me about during the biopsych lectures, if we were going to cover synesthesia, we'll cover that during this lecture. Probably not get to it till next week. But yeah, so there's lots of cool topics here that we can cover. So just making sure I'm not missing anybody real quick, and then we will get started. Okay. So I'm going to go ahead and share the PowerPoint. And I'm going to just give you a heads up before I screen share that the background for this is a giant eye. So just like, it's thrown people off sometimes. So just FYI, you're about to see a giant eye. So I thought it was appropriate, but it has also like freaked some people out at times. So I apologize if it does that to you. So get myself set up here. And for folks who are just rolling in, I just let everybody know that we'll do the Explain It to the Media quiz on Monday instead of today, just because you just took the exam. I should have the exams graded by Monday in theory, unless something goes horribly wrong. And so then I'll be able to tell you everybody did. So, but at least logistically it worked. So that was good. That was like the bare minimum we were trying to get going, right? So. I'm going to pull up the chat. So as per usual, if you have any questions, comments, please go ahead and type them there. All righty. So sensation and perception are two terms that are sometimes used interchangeably in sort of lay psychology vernacular. but in fact, they're not the same thing at all. So I think it's important that we start with definitions for the two terms. So sensation is a process by which a stimulated receptor creates an internal pattern of nerve impulses that represent the stimulus in the brain. And that's really wordy, so I'm going to say that a couple more times, so no worries. So it's a process by which a stimulated receptor, internal pattern of nerve impulses, internal pattern of nerve impulses that represent the stimulus in the brain. So a process by which a stimulated receptor, could be an eye, could be an ear, could be some sort of receptor in your fingers for touch, creates an internal pattern of nerve impulses that represent the stimulus in the brain. So it's basically how outside information gets to our brain. Perception is the process that makes those sensory patterns meaningful and more elaborate. So perception makes those sensory patterns meaningful and more elaborate. It basically, sensation is how we get outside information to the brain. Once it's there, perception is how we understand it, essentially. It's the brain converting things and applying extra meaning to them so that we know what's going on. And this will become much more apparent as we go through examples with the specific senses. But so for now, just sort of remember, sensation's taking information from outside of us, bringing it into the brain. Perception allows us to essentially translate and elaborate on it. So the purpose of perception is to take that information from the outside world and represent it internally in a way that makes sense. From an evolutionary perspective, the purpose of perception is to adapt to stimuli in order to survive, right? So example, we see a bear, right? If our perception doesn't include, you should probably think about trying to get away from the bear, right? Then evolutionarily, we could end up not being able to procreate because we're dead, because the bear got us, right? And other things as well, right? So being able to recognize, someone who might be attractive to you, who you might want to mate with in the future, things along those lines. How does stimulation become sensation? The brain senses the world directly because of these sensory receptors. We'll come back to those in a second. So the sense organs convert stimulation into the language of the nervous system. So to neural messages. Essentially, it converts this outside information to action potentials, okay? And then those get translated across the neurons through all the neurotransmitters we just talked about. The senses all operate pretty much the same way, but each extracts different information and sends it to its own specialized processing region of the brain. Some of these are more obvious. We talked about how the occipital lobe does all the visual processing, but some of those are a little less obvious, and so we'll kind of talk about how those things relate. So there are these are examples of our types of sensory receptors, and for what our various senses look like. They look kind of weird, right? They look like little amoebas or something, right? But they're really important, obviously, because they're what translate, again, this outside information to things that we can interpret. Yes, Lara, go ahead.
Do we have to memorize how those things look?
No.
For each of the senses?
I'm never going to show you a picture.
I'm sorry, I couldn't hear that. Say that again?
I said no. I wouldn't have a diagram and have you label them. Just sort of
okay.
what each does, essentially. Essentially, we'll go over the type of thing. You won't need to know what the picture does. Alrighty. So good question. Yes. So I just want you to know essentially how these translate the information to the brain. And that's what we'll spend the majority of this lecture talking about. So sensory receptors are cells that detect and transmit information to the sensory nerves in the brain. And they set off action potentials in the sensory neurons, which then carry the information to the central nervous system. Actually, I got a question in the chat. ICE have two. Actually, ICE have a lot more. ICE have a ton of them. We'll talk about it when we get there. All of our senses have a lot of different types of sensory, but yeah, there are two types of visual. Maybe that's what you're asking. Two types of visual receptors. So there are the rods and cones, if you've ever heard of that. And so we'll talk in depth about what those different things mean and what they do. So yes, eyes have two types, but lots of different receptors for each type of sense. Let's see. The sense organs and sensory receptors have three classes. This is what I was starting to say at the beginning of the slide, but I was getting ahead of myself. So I'm going to type the names of those real quick for you. So the first type is photoreception. Let me make sure this is going to go to everybody. Lara, can they give the wrong signals? Sort of, right? So they're never, like, your photoreceptor is never going to give you like another type of signal, but it's possible for the eye to be tricked. So think about the very first day of class, how we looked at those optical illusions, right? We thought things were moving. Also, if you do have any kind of injury or brain problem or eye injury, it's entirely possible that they could just not give any information to the brain or be firing with incorrect information. So yeah, really good question. And obviously that's true of the other senses as well, right? So if you are hard of hearing, it's possible you don't get the information you need, and there's some brain disorders where a certain part of the brain is damaged and you can actually hear things inaccurately or not be able to translate that to be able to respond to it. So that's a really good question too. Okay, so the first type of sensor is photoreception, and that involves the detection of light, and that's only vision. Okay, so vision is entirely photoreception, and you have these two types, the rods and the cones, that view that. Next is mechanoreception. And so that is the detection of pressure, vibration, and movement. Pressure, vibration, and movement. And that's perceived as touch, but also as hearing and things like equilibrium. So your receptors for hearing actually operate based on mechanoreception. And we'll talk about the ear in much more detail, probably starting Monday. Usually the eye takes almost a whole day. And essentially what happens is sound waves, the vibrations, are turned into essentially mechanical things by the hearing receptors. And it's detected that way, essentially as vibrations rather than sound per se. Then the third class is called chemoreception. And as the name sounds, it's a detection of chemical stimuli. And so smell and taste both fall under this. So when you're smelling, you're actually taking in teeny particles of chemicals, basically. Chemoreceptors, and taste does that as well. And here, one thing I want to note, when you hear me mention chemicals, some people immediately think of horrible toxic things, but just remember, chemicals are what make up our bodies, right? Like the largest chemical or the most common chemical in our body is dihydrogen monoxide, which is water, right? That's a chemical. So I think people in our society sometimes hear chemical and think something horrible, but like all living things are made up of chemicals. So. The next thing we're going to talk about is psychophysics, which is the study of linking physical properties of stimuli with a person's experience of them. And one of the ways we talk about this is through thresholds. So again, psychophysics is linking physical properties of stimuli with a person's experience of them. So as I just mentioned in response to Lara's question, yeah, sometimes we can misperceive things, right? Our eyes can trick us, things like that. There are things we can't detect. So if you think of something like a dog whistle, right? Dogs can hear it, but we can't because it's not within our range. Things along those lines. So when we talk about thresholds, the first thing we talk about is absolute threshold. And this is the amount of stimulation necessary to detect a stimulus. The amount of stimulation necessary for a stimulus to be detected. So, you know, how loud does something have to be for us to hear it at all? So think about when you maybe have muted your phone, right? Because you didn't want to listen to an ad, right? And then when you go to turn it back up, it's like, how many little bars do I have to have in order to actually be able to hear it properly? So that's the absolute threshold. So it's the minimum amount of energy needed for a person to detect something. So whether that, again, is like light energy, mechanical energy, vibration, touch, or chemical energy, right? And if something falls below the absolute threshold, then the person just doesn't detect it. They don't experience it, even though it's happening around them. Under ideal circumstances, a person's absolute thresholds are very low, right? We want to be able to have a pretty big range of what we can detect. The problem is there's so much noise around people that they cannot detect half the stimuli that are confronting them. And so noise here doesn't just mean auditory noise, it means extra stuff, right? So if I were to try to pay attention to everything that I could sense right now, right? I'd have to pay attention to my feet on the floor, what it feels like to move my hands through the air, what it feels like to be wearing a necklace, to be wearing a watch, to be wearing clothing, right? What it feels like to have my hair touching my face, what it feels like to hear the fan in the background. And then in terms of seeing, you know, I have both my monitors, I have my notes, I have a window over here, right? I can't pay attention to all of that simultaneously. It's just not humanly possible. And so our brains quickly learn to focus on what's important, right? So noise is the term given to these irrelevant and competing stimuli that we can then sort of tune out so that we can pay attention to what we want to. Yes, Nick, this is a really good question. So why animals stand motionless to hear better? Yes, and this is also why if you are driving to a place you've never been before and you're looking for the address, you'll find yourself turning down the radio, right? Like it shouldn't actually matter if you think about like sound versus sight, right? But it allows you to focus more on what you need to focus more. So yeah, really good questions. Yes, this is what happens when animals might stop so they can hear, so they don't hear their own rustling, right? Or again, we do something like turn down the radio so we can actually find that address. So then there's what's called the difference threshold. And this is the smallest amount by which a stimulus can change and we can detect the difference. So the smallest amount by which a stimulus can be changed and the difference be detected. And this other term underneath it, the just noticeable difference, just means the same thing. So the difference threshold and the just noticeable difference are the same thing. Just two terms for the same thing. So it's the smallest amount by which a stimulus can be changed and the difference be detected. So this could be difference in sound levels. This could be difference in shades of pink, say, right? How much does it have to change until we can tell the difference? I believe Lara had her hand up.
You actually just asked the question for me. How much does it have to change?
It depends on your sense. It depends on what we're dealing with, right? That's a really good question. Yeah, it really depends on a whole bunch of other things. How much is going on, right? So if I'm trying to notice the difference in sounds in a very quiet room, right, that's really different than if I'm doing it at like Times Square in New York City, right? So yeah, this can really vary depending on what you're trying to sense and what the other things around you are going on. So related to this is Weber's law. And Weber's law says that the size of the just noticeable difference, the JND, is proportional to the intensity of the stimulus. So this also goes towards answering Lara's question. So the just noticeable difference is large when the stimulus intensity is high and small when the stimulus intensity is low. So Weber's law basically says that two stimuli must differ by a constant minimum percentage rather than by a constant amount. So it's not like, you know, X decibel to X decibel. It's like this percentage of it kind of based on everything else going around, right? So something's really obvious, really there, you know, maybe, again, I'm trying not to just use sight, think about like touching something, right? And you can tell the difference, right, between whether something is burning you hot or just kind of warm, right? Well, there's a percentage in between that lets you figure those out. Or if you've ever like felt a child or a sibling's forehead to try to figure out if they have a fever, right? If you do that enough, you start to like tune that ability. So I'm pretty good now at telling when Quinn has a low-grade fever versus when she's just sort of normally warm. So yes, those are the just noticeable difference. And again, Weber's law says that they have to differ by a constant minimum percentage rather than by a constant amount to be perceived as different. So here are some of the absolute sensory thresholds for most humans. Now, granted, there are people who are, you know, better attuned to some things, and so this might vary. There are also people, obviously, who have deficits in these senses, and so this would vary for them. But vision, a candle flame at 30 miles on a dark, clear night. Well, that's pretty hard, right? Hearing a ticking watch at 20 feet under quiet conditions. And if you've ever had a ticking watch or clock in your room when you're trying to fall asleep, this might resonate with you, right? Until I got my Fitbit, I used to wear a watch that ticked, and I actually had to like be careful how I set it on my dresser at night. Because if I set it a certain way, the tick just like, I couldn't sleep with hearing it. Smell, 1 drop of perfume diffused throughout 3 rooms. And again, this will be affected by a whole host of things if you're stuffed up, all that type of thing. Taste a teaspoon of sugar in 2 gallons of water. And again, you're stuffed up. This is going to be a problem because part of taste is smell. Touch, the wing of a fly falling on your neck from a distance of 1 centimeter. So if you've ever been like, ooh, and there actually is a bug there, right? You've sensed this. But again, this is going to differ based on, you know, are you having some sort of sensory issue, right? Is your arm asleep? Do you have hypothermia? Are you wearing clothes even, right? Like if I have a turtleneck on, that's going to be different than if I'm wearing something like I'm wearing right now, right? So all of these things are like, in theory, the absolute sensory thresholds, but these are under ideal conditions. One of the things psychologists got really interested in the 50s, especially the 1950s, was the idea of subliminal perception. So this is the idea that there might be information below the level of conscious awareness that could still influence us. That even though we didn't realize we could detect it, we still were. So the most widely known piece of research into subliminal messaging comes from a market researcher whose name was James Vickery. And his study is perhaps the most famous as it was one of the first of its kind. It was a large-scale investigation into the power of subliminal messages. And it was conducted on the public, and it had some alarming results. So he did this study in 1957. He used a movie theater in Fort Lee, New Jersey. And over a 6-week period, he tested subliminal messaging on over 45,000 moviegoers. Now, interestingly, this was like before IRBs and things like that, so he wasn't like having them sign informed consent when they went to the movies, right? So while the patrons watched a movie, and the movie was called Picnic, if anyone's curious, he displayed 2 subliminal messages. One stating, "Eat popcorn," and another one, which is why this is what's on the screen, and the other one saying, "Drink Coca-Cola." The messages were text-based subliminal messages, and they were displayed much faster than we think the human eye can see. So for 3 1,000ths of 1 second, 3 1,000ths of a second, You just got to try to wrap your head around that. And they were displayed once every 5 seconds. Results were taken by comparing the current 6-week sales of Coca-Cola and popcorn to the sales figures from the previous 6 weeks. And the difference was noticeable. So popcorn sales rose by 57%, and Coca-Cola sales rose by 18%. That sounds really cool, but Vickery lied about his results. So he was asked to repeat the test and found no significant increase in sales, and eventually he confessed he had falsified or made up the data in his first study. Wah, wah. So yeah, don't falsify data, that's bad, right? And perhaps subliminal perception is not as much of a thing, or at least not as influential as we thought based on that study. So Strahan decided to try to play with some of this in 2002. It was a study of thirsty versus non-thirsty words. So he had participants not eat or drink anything hours before they came in, and then code words on a computer. So the experimental group was subliminally primed with thirsty words, so words about drinking or wetness, water, things like that. And the control group was neutral. And these were shown in a specific parts of the screen for 16 milliseconds, or thousandths of a second. So not the 3 from the Vickery study, but 16 thousandths of a second, which again, still not very long at all, right? A participant who received the thirst prime drank significantly more liquid than the participants. So what happened is participants came into the lab, they sat in front of the computer. Again, they had been asked not to eat or drink anything for 3 hours beforehand. You can go 3 hours without eating or drinking. You're okay, right? They're not like starving these people. They saw these words and they were like sort of directed to a prime. They didn't realize they were seeing them because they were so quickly displayed. But the people who, and then afterwards they were given something to drink. I'm guessing probably just water. And the people who got those subliminal primes for thirst consumed more water afterwards. So maybe there is something to this. And again, we talked about this with the idea of blind sight with people with their corpus callosum cut, right? Like they can't actually name what they're seeing, but if they are asked to draw it, they can actually draw it. So we know that the eye and the brain can process things that we don't even realize we're seeing. And this is an example of that again in like this really quick method. So not as strong as Vickery actually originally said, but according to Strahan, maybe some stuff going on here. Another thing that comes into play here is what's called signal detection theory. So, signal detection theory focuses on decision making about stimuli when you're in the presence of uncertainty. So, signal detection depends on the individual and the context, just like I've been saying with all these other things. And the context here can include things like how tired you are, so fatigue. It can include expectancy, so do you expect to see a stimulus, or hear a stimulus, or feel a stimulus, right? And it can also affect on the urgency of the moment, like does it make a difference in the moment? So the decision is, did I detect something? Did I actually hear something, see something, touch something, taste something, smell something? So information acquisition refers to the information that's produced. Criterion is the basis for making a judgment about the available information. And then there are 4 possible outcomes. So there's the hit, miss, false alarm, and correct rejection. And I have a whole chart about those on my next slide. Are you guys having internet problems again? Ugh. Okay, well, I am recording this. This will go up on YouTube. So If you miss any piece of it because the internet is sucking, it will be available for you. So yeah, I think we really are just experiencing everybody's trying to be on an online class at once, a remote class at once. So that's what creates the problems we see. All right, so signal detection theory. So we're looking at, did the signal actually occur? Was something actually presented? And how did the observer respond? So let's say we're trying to do a visual stimulus. We flashed it up there for 16 milliseconds, right? Maybe a little longer. And then if the participant says, yep, I saw something, and there was actually the signal present, then that's called a hit. It means correct. You hit it right on the nose, perfect, right? If there was a signal present and the person said, no, there was nothing there, then that's a miss, that's a mistake. If there was no signal and you said, yes, there's something there, then that's a false alarm. And obviously that's a mistake as well. If there's no signal and you say, nope, nothing's there, then that's a correct rejection. So basically there are two ways to get it right. If there is a signal present and you say, yep, I saw something, or if there's not a signal and you say, nah, I didn't see anything. But if you say, no, there was nothing there when it was, or if you say, yep, there's something there and there wasn't, then that's where you can end up with these misses or false alarms. So he's obviously not the best example for anything anymore. I don't really like to use Bill Cosby as examples for things, but he had a bit he did, I don't remember if it was on the Cosby Show or in his stand-up, but where he talked about doing the hearing test at the doctor's office where they put the headphones on you and then you hear the different beeps, right? And he was worried about being tested for hearing loss. And so he just started raising his hand constantly, right? Like, oh yeah, there's something, there's something, there's something. And so he probably had some false alarms because there were probably some times where there was not something there, but he was saying he heard it, right? He was trying to eliminate the misses, the mistakes, but in so he was committing a lot of false alarms. So this becomes really important in terms of detecting if someone is having some sort of sensory problem, right? It's also really important in research because this is a lot of the ways we report things when we are doing research around sensation perception things. All right, so this is called the Stroop. And I think this would be nearly impossible to try over Zoom. So I will be the guinea pig here, okay? So what I'm gonna do is I'm gonna start with the left-hand side and I am going to say the color of the ink cubes. And this might turn up slightly different on your screen. Sometimes the yellow looks orange for y'all. I'm gonna say yellow, okay? So yellow, red, green, blue, red, green, blue, yellow, green, yellow, red, blue. Pretty easy, right? Now what I'm gonna do is I'm also gonna keep naming the color, but I'm gonna do it with these words. So red, blue, green, yellow, blue, red, yellow, green, red, blue, right? Okay, so this is called the Stroop effect. And this is something also we use a lot in psychological research. This can also be used as a diagnostic tool. Basically, what we have to do when we're doing the one where we're reading words, we want to read the words, right? That's what we're trained to do from a pretty young age, is to read the words. But when we have to just say the color, and the color doesn't actually match what we're used to reading, right? That's where this becomes something where you have to inhibit your normal response. So if you remember, we were talking about the prefrontal cortex, what Phineas Gage had injured when we were doing biopsych. So that is involved in inhibition, sequencing, executive control. And so you have to be able to use that in order to do this inhibition, right? Otherwise, you're just going to read the words. And as you can see, I don't know how noticeable it was, but it slowed me down, right? I was able to name just the colors pretty quick, but when I had to inhibit reading the words, I was slower at things. So you can actually use this diagnostically in a clinical neuropsychological assessment to help you assess whether that person is impaired on this part of the executive functioning, impaired in their ability to inhibit those natural responses. Now, one of the, or I guess they're learned responses, come out of the womb, able to breathe, right? Now, one thing that's really important is obviously when you're using the Stroop, you have to use it with someone that you know has the ability to do it, right? So you couldn't use this with someone who's colorblind. You'd have to use a different test of executive function. You couldn't use this with someone, who might have an eye injury, right? That it doesn't let them see all of what's going on, or some sort of brain injury. So, you need to use this ethically and with people who can use it, but it ends up being a nice quick way to do an executive function screen. Alrighty, so as promised, we are going to start with the eye. So the eye does visual sensation, as I mentioned before. It's doing photoreception, it's bringing in light and interpreting it. And so the eye is set up like a camera that gets pictures of the world. And it's trying to get the best picture of the world. And the way it's set up is really meant to do this, although again, obviously there are cases, honestly, I think sometimes for more people rather than not, where this might need some correction, right? Off, and so you need glasses or contacts or something like that. So the sclera is the outer part of the eye, though it's the white part. If you're looking at your eye, it's the white part. Try not to poke myself in the eye here. And it gives the eye its shape, and it protects the eye from injury. So that's the sclera, which I think, now that I'm looking for some reason, is not depicted on this picture, but it's the white part of the eye. Again, the iris is here. That's your colored part of your eye. So whether you have blue, brown, hazel, green, I don't know. I think some people have like violet eyes, things along those lines. That is determined by the color of your iris. The pupil is the black part of your eye. Okay, and so the pupil is this piece here, right? And it's the opening in the center of the iris. So the iris, the part of your eye that's colored, actually contains muscles. And those muscles allow the pupil to get larger or smaller. So if you've ever wondered, how does that happen? And especially if you have a cat, so like our cat, when he's about to pounce, his eyes get, his pupils get really, really big, right? How does that happen? Well, it's actually a muscle in the iris of your eye that allows it to dilate or to come back down. So the iris contains the muscles that allow the pupil to get larger or smaller, and it does that in reaction to how much light is being let into the eye, or how much light it needs to let into the eye, like when my cat is trying to bounce. The cornea is the clear membrane on the outer part of the eye. So it's the part that if you wear contacts, you put your contacts onto, basically. I'm trying not to poke myself in the eye here. The curved surface on the cornea bends light on the surface of the eye, and it does that in order to get it to focus on the back of the eye here, okay? So the cornea is this piece here, okay? And so light passes through there, and it's like a lens. So if you have, actually, I have my glasses right over here. Okay, so like if you have glasses and you look at them from the side, mine aren't super thick, but they have a bend to them, right? And so essentially what your glasses are doing is the same thing your cornea is doing, only they're correcting what your cornea is not doing right. They're helping to focus the light on the eye. So the lens is transparent and somewhat flexible. This is the lens here, this big round thing. I guess it's kind of ovaloid. And when a person's looking at an object far away, the lens has a relatively flat shape. However, when a person is looking at an object that is closer, it'll curve to bend the light to help us focus on it. So, getting more into the parts of the eye in the back of the eye here. So just to orient you real quick, based on the diagram we were just at, we're not going to be looking back here. Okay, so we've been talking all about up here. Now we're going to be looking back at this part of the eye. Okay, so the retina is the light-sensitive layer at the back of the eyeball. And it records what a person sees and converts it into a neural impulse for processing in the brain. The retina is the most important mechanism for sight because it is the one doing this conversion of the photo, the light, right, into essentially action potentials, things the neurons can pass on. Photoreceptors are light-sensitive cells in the retina, so I think they're represented by the little green dashes here, although there's certainly more than that, that convert light energy, there we go, into neural impulses. So they're the parts of the retina that do this work. So as I mentioned, there are two types, rods and cones. Rods are sensitive to dim light, but not to colors. And cones are sensitive to colors, but not to dim light. And this is pretty easy to remember, just see color, see cone. That's how you can tell the difference between rods and cones if you're trying to remember it for a quiz or the next test. And then the fovea is the center of the retina, and that is our area of sharpest vision. And interestingly, the fovea only has cones, it doesn't have rods. And it is important for many visual tasks. So again, real quick, I'm going to pop back to the previous slide so we can take a look here. This does show you the fovea here, that little piece, that's the area of biggest focus. Again, here is the retina, and the rods and cones would be throughout the retina. So this is why there are two different types of photoreceptors, two different types of receptors in the eye. One is doing dim light, one is doing colors. So this is why, like if you're out at night looking at things, everything will kind of seem like it's in grayscale if there's not light around, because you're using your rods at that point in time. So how do we change the light to these neural impulses, these action potentials? So light comes into the eye. The light here is represented by these yellow long arrows, which again might look orange on your screen, but they're yellow on mine. And it goes to the back of the retina. And it passes through the ganglion and bipolar cells before it strikes those photoreceptors. And again, the photoreceptors are the rods and cones at the back of the eye on the retina. So once stimulated, the rods and cones convert the light information and translate it to the bipolar cells. So they get the light comes all the way back here, and then it's interpreted by the rods and cones, and they then transmit it on to these things that are purple on here. They're not purple in real life, that are called the bipolar cells, okay? So the bipolar cells essentially collapse the information from several rods or cones, and then they transmit it to the ganglion cells. So you can see each of the bipolar cells is talking to several different rods and cone cells. And then each bipolar cell talks to one ganglion cell. And the ganglion cell is what then transmits it to the brain. So the axons of the ganglion cells are what make up the optic nerve. So if you've ever heard anyone talk about the optic nerve, If you go to your eye doctor, and now more and more, instead of doing the thing where they dilate your pupils and whatnot, they can do a thing where they just take a picture of your eye, and they'll be like, "Oh, that's where the optic nerve is." So what they're talking about is the axons of the ganglion cells. So the impulses travel from the ganglia to the brain via their axons that make up the optic nerve. So again, we've now converted in essentially 3 steps light energy to something that the neurons in the brain can interpret. And just again, to orient you, we're looking at this part of the brain, or the eye, sorry, right here. So right here at the back. So the optic nerve, again, the axons of the ganglion cells, the bundle of the neurons that carries information from the retina to the brain. So again, we're looking at the ganglion cells, their axons, carrying that information back to the brain, and we call that the optic nerve. We also have what's called a blind spot in our eye. And this is where there are no photoreceptors. There are no rods, there are no cones, because it is the place in our eye where the axons of the ganglion cells, where the optic nerve actually exits our eye. So this is our blind spot. That's the optic nerve where it exits the eye. So we actually do have gaps in our visual field here, but you don't experience blindness here because the other eye fills it in. So basically whatever one eye isn't able to see, the other eye can fill in. So it's not like we see little blind spots in front of us, right? Our eyes are able to give us a continuous picture till about here. And again, in different animals that might be different. Animals that have their eyes on the side of their head and kind of see a lot more behind them than we're able to. Those tend to be prey animals sometimes, so that they can kind of watch out for predators behind them. So that is the blind spot. It's where the optic nerve exits the eye. All right, so I mentioned blind sight, and we talked about this a little bit when we talked about, again, people who have their corpus callosum centered. But blind sight can happen in people who don't have that happen. Now, this is a phenomenon in which people are perceptually blind in a certain area of their visual field. So maybe, it could be the whole visual field, or maybe they just can't see from here over. That can happen. And so they will demonstrate some response to the visual stimuli presented in that part of the field, even though they say, I can't see it. They can still respond to it in some way. So for example, let's say you present a green ball in that patient's visual field. You ask them if they see a green ball, and they'll be like, no, I don't see anything. And then you say, okay, point to the green ball. And they're like, no, I can't see it. How can I point to it? And you're like, just humor me, point to it. And they'll actually point to exactly where you're holding the ball. So this is the phenomenon of seeing things that you don't know you can see. And this is called blind sight. So again, they can have no conscious awareness at all of that stimulus, but they're able to predict at levels significantly above chance aspects of a visual stimulus. So is it round, is it square, location, again, they can point to it, type of movement, is it moving left and right, is it moving up and down, right? And we can do this with a forced response, so sort of like a multiple choice, is it moving up and down, or is it moving left and right? And again, they'll be like, I can't see it. You'll be like, humor me. And they'll be like, left and right. And they'll be like, yep. Okay, so part of their brain is processing this, but they don't realize it. This can also occur when subjects have some awareness of, for example, movement within the blind area, but no visual percept. So this can be caused by, for example, a person being aware of their eyes tracking motion, which will function normally. So it's sort of like they recognize there's movement because they can feel their eyes moving, rather than feeling like they could see the movement. So blind sight is often caused by injury to the occipital lobe, the part of the brain responsible for vision. And it's a significant phenomenon of intact brain function as well. So blind sight and other similar phenomena in other spheres, so, essentially blind hearing, even though that's a weird term, right, is an important ingredient for a variety of activities where one wants to move quickly and appropriately without thinking about it, right? So if you play a sport, right, you don't have to think about, well, now I do this with the ball, now I do this with the ball, right? Your body just kind of like learns to do it, right? And the similar phenomenon to blindsight is what takes place here. So I'm going to see if my hyperlink is still working. Maybe, hopefully. Alrighty, yes, I have a video about blindsight I'm going to show you all. So I will share that portion of the screen. So I had to pause the video. Make sure I'm sharing the sound for y'all. Okay, and start the video at the beginning.
You're now, I would claim you're now awake, you're wide awake, and you can be aware of my face, of the fact what sort of, that I wear a colorful shirt, that I talk with this Schwarzenegger-like accent. And so this I would call an operationalized definition of consciousness.
Christophe, why have you and Francis Crick, who I guess is best known for discovering the structure of DNA, chosen visual awareness to study consciousness?
We believe that because consciousness is an evolved property that probably nature discovered, it was discovered once during evolution, and the different forms of consciousness, i.e. visual consciousness, auditory consciousness, maybe even high-level self-consciousness, are probably just different variants of the same theme, the same mechanism.
So they've designed experiments to test visual consciousness. Eileen Wexler has normal vision, but this test simulates a phenomenon called unconscious vision, or blind sight, that can occur when there's damage to the brain's visual cortex.
So the world to them looks like this. They're blind in the left side of the visual field. And they go to the doctor, and the doctor says, well, can you see this? No. Well, can you guess whether it moves to the left or to the right? And the patient says, well, I'm blind. And the doctor says, well, guess. And the patient guesses left, right, up, down. And the patient is as surprised as the doctor, at least they were at first. And the point is that these patients, there's now a quite substantial number of them throughout the world, they do not have conscious access to information in their blind hemisphere. Yet, if forced to respond, it turns out that something in their brain has information because they can correctly guess, for example, the direction of motion. They can correctly, roughly point at a bright light. They can roughly say something about what colors is out there. No, it's very crude.
Glasses. Senior research fellow Jochen Braun designed these blindsight experiments.
So let me show you the image one I will see first. Okay, that's what it looks like. You have a background of squiggles all aligned. Yes. And there's this little area where the squiggles, they point at a right angle. Yeah? So I'll be asking you, where is this area, this different area?
It sounds easy, but during the experiment, the only thing I'm conscious of is a uniform pattern. Even though my left eye sees this, and my right eye sees this, my brain superimposes them. The theory is that some part of my brain will still know how to find the target, even though I'm not aware of it.
Now, the reason we do this is that the primary visual cortex, the first level of the brain which receives information from the eyes, receives information from left and right eye separately. So at this level, the brain can still figure out where the figure is.
Because it hasn't been superimposed.
It hasn't been superimposed, but at the next level in the brain, it's superimposed. So at that level, the brain is blind to where this figure is.
The glasses make sure that each of my eyes will see independently. Okay, here we go. I feel a bit foolish. I can't see any variation in the pattern. It seems impossible to find the target.
When we designed this experiment, we didn't know what would happen. And then the student who did the experiment came back and said, this is very strange. I'm rather good at finding the square, but I don't see it. So then it dawned upon us that this information, which does make it to the brain, at least at the first level, you can still use it, but you're not aware of it.
The results haven't been consistent, but Aaron Rosen is one of the students who can sometimes locate the target 70% of the time, even though he's not conscious of it.
The theory, which has been advanced by others, not only by us, is that there are actually two visual systems, one ventral and one dorsal visual system with different functions. So the function of the ventral visual system would be to recognize things, to read, to see faces, to acquire visual memories, and so forth. And that visual system, the theory goes, contributes to your conscious experience. The other visual system, the dorsal one, has a different function, namely to guide your movements, your eye movements, and also your hand movements, or just locomotion if you're walking around.
Or say reaching for a cup of coffee.
Exactly.
Or a glass of beer. Here's another way of looking at the two kinds of vision, conscious and unconscious. No matter how you view this glass of beer, you know it's a glass of beer.
Over to the side.
But to pick it up and move it around, a separate visual system kicks in.
In some sense, we have within us a kind of robot that allows us to interact with the world that is controlled to some degree by our conscious visual experience. So our conscious visual experience marks this as a goal object, and then we bring to bear a robot that allows us to reach out and pick this up. Different computational demands.
So in a sense, that's unconscious vision.
It's unconscious vision, and it can, in some real sense, never be conscious. It's quite inaccessible to conscious report.
Okay, so I'm going to reshare the PowerPoint here. Better pause that. All right, so that's blindsight. Yeah, and so we all have an element of this, and it is a thing, or we can not be aware that we're seeing something, and it's actually being processed by part of our brain. So this diagram shows how visual information is processed once it's taken by the axons of the ganglion cells to the occipital cortex. So the lens of the eye receives the images and puts them onto the retina. The visual system splits the retinal image from each eye. And I like that they color coordinated this. Again, it's not this way in real life, but you can see everything coming from the left eye is yellow here. Everything from the right eye is sort of this purpley blue, right? And then what you'll see is everything on the right part of what each eye sees goes over to this part of the brain. And everything on the left part of what both eyes see goes to this part of the brain. So your brain is taking the information from both. So this part right here, called the optic chiasma, this is the other part of the brain where messages can go across the two hemispheres. So if people do have to have their corpus callosum severed, they can still do some communication across hemispheres via the optic chiasma. So, sort of, again, tying everything together from last lectures to this lecture. So part of the image from each eye crosses over to the opposite side of the brain at the optic chiasma, or sometimes it's just called the optic chiasm. And objects in the left part of the visual field are both eyes are sent to the right hemisphere of the visual cortex in the occipital lobe, and objects from the right part of the vision from both eyes are sent to the left hemisphere of the visual cortex in the occipital lobe. So our brain sort of like flips things around for us once we get it there so that we can actually see things as they are occurring rather than a mere image. So when we were talking about the just noticeable difference, there are, properties of light that allow us to detect it, or again, might mean it's outside of our ability to detect it. So one is wavelength, and that's the distance between peaks. So this is perceived as hue, H-U-E, not some guy named Hugh. And some wavelengths are beyond human sensation. Amplitude is the height of the wave. So again, if we're thinking about just a curve that goes like this, wavelength is the distance between the two peaks. Amplitude is how high that curve is, right? Does it go like this? Or does it go like this, right? And amplitude is perceived as brightness. And then purity is mixture of wavelengths, and it's perceived as saturation, particularly of color. So there is something called the electromagnetic spectrum, and this is the entire range of electromagnetic energy. But we can only perceive the visible spectrum. So the electromagnetic spectrum includes things like radio waves, X-rays, microwaves, visible light. But we can only see the visible light part of that. It's a tiny part of the electromagnetic spectrum that is visible to our eyes. So here is where we can start to look at that. So here is the entire electromagnetic spectrum on top, all the way from gamma rays to like past AM radio, right? And microwaves are in there, ultraviolet, infrared are here as well, X-rays, TV, things along those lines, right? So we can only see this teeny tiny portion right here. And that's our whole Roy G. Biv, if you learned that in school, red, orange, yellow, green, blue, indigo, violet, right? So we can only see a teeny portion of the full electromagnetic spectrum, okay? And this is what's called the visible light. And here you can see the idea from wavelength, that they get shorter or longer as we go across this spectrum. And so how do we see these colors? Well, there's a couple different theories. So trichromatic theory is based on the fact that we have 3 different kinds of tones. And this actually throws some people off because we just sort of assume, oh, it's the primary colors. But no, actually, the 3 types of tones are red, green, and blue. So not red, yellow, and blue, red, green, and blue. Those are our 3 types of tones. And so initial stages of color vision is what this explains, essentially, right? So when it hits those rods in the retina, that's what this is looking at. So right when the light comes in, it hits the rods. Maybe that explains why we see color the way we see it. It doesn't explain everything, though, about how we see color. So there's also what's called opponent process theory. So opponent process theory is based on bipolar cells, and it explains the processes there and beyond. So they say that the colors are in complementary pairs. So like red and green would be a complementary pair. So this is why, for example, for those of you who wear makeup, if you've ever bought certain types of concealer for blemishes, right? For red blemishes in particular, they're green because they balance out and neutralize the color. So this explains after images, which are sensations that linger after the stimulus is removed. All right, so let me, yes, okay, good. Just making sure my PowerPoint was right. Okay, so what I'm gonna do is I'm going to click the next slide and have you focus on it for 30 seconds. Now I'm gonna time it so that we make sure it's actually 30 seconds. So give me a sec to get to my timer. Okay, so here we go. You just stare at the dot in the middle of the image for 30 seconds. All right, now stare at this slide. In theory, you should see the essentially after image of the last slide, and it should be the American flag in the normal colors. If you don't see it, don't worry, you don't have something wrong with your eyes. It's a little harder to do on a screen than it is to do normally, but some of you may have seen that, and that's what an after image is. And this is based on the fact that we have this is one of the bases of the idea of opponent theory, opponent process theory. So honestly, like a lot of theories in psychology, there are probably, oh sweet, at least one of you saw it. So some of you saw the shape, not the color, cool. So there are probably correct components of both of these theories, honestly. So I think that that's important to remember as we talk about different theories for different things in psychology. No one theory is usually totally correct, right? There's always sort of elements of truth and not truth to other things. All right, so another thing that can happen is color blindness. So this is a vision disorder that prevents an individual from discerning certain colors or discriminating certain colors. The term colorblind refers to seeing some colors but not others. Color blindness depends on which of the three kinds of tones is not working. Right, so remember you have red, blue, and green, and if one of those isn't working, it makes it harder for you to see certain colors and not others. So you don't have to like out yourself if you don't want to, but there may be some of you in the room who cannot see the number, right? So the number here is actually 29, but if you are colorblind, you might not be able to see that. Also, you might not be able to see that depending on the size of your screen. So just put that out there, like it might not be color blindness. Yeah, Lara, I think you had your hand up.
Never mind, I got answered.
Sweet. So in theory, I have a video on color blindness. Let me see if it works. Nope. Okay, that's fine. We will skip it. We are good to go. So again, this is something we need to be aware of that can affect your perception, obviously, because it affects your sensation. So visual perception is organizing and interpreting visual signals. So there are a whole bunch of different dimensions, and we're going to have slides for each of these. So don't worry about jotting these all down right now if you don't get them. Each will have their own slide. So these include shape, depth, motion, and constancy. So we'll start by talking about shape. To perceive a visual stimulus, fragments of information that the eye sends to the visual cortex must be organized and interpreted. So one of the things that we look at here is the figure and ground relationship. And this is part of Gestalt psychology, that the idea here is perceptions are naturally organized according to certain parts, and that the whole is different from the sum of the parts. And this might not make that much sense, but there's a story that I've actually heard in several people use in different speeches or sermons throughout the years. There was a group of blind monks, and they brought them to an animal, and they had them touch different pieces of it. And one of them said, "Ooh, I think this is a snake." And one of them said, "Ooh, this actually feels, maybe like the trunk of a tree, actually." And one of them said, "Oh, it feels like, maybe a ray or some sandpaper." And one of them was touching the trunk, and one of them was touching the ear, and one of them was touching the leg, right? You'd feel a tail, it might feel like a rope. And so the idea is, when we put everything together in the Gestalt, we can start to figure out, oh, that was an elephant, right? But in those different contexts, if you only get a part of the thing, you might not be able to figure that out. So Gestalt psychology explains how people naturally organize their perceptions according to these certain patterns. So the figure-ground relationship occurs when a person organizes the perceptual field into stimuli that stand out the figure. For humans, this is most often like another human, to be honest, although it can be an animal or a building that's really important, right? And those that are left over. And whatever's left over, whatever's kind of in the background or we're not focusing on, that's the ground. So figure is what we're focusing on, ground is sort of everything else. Closure is a Gestalt principle which says that if we see a disconnected or incomplete figure, essentially we are able to close it in our mind and we see a whole. Proximity is a second Gestalt principle where individuals see objects as close to each other, they tend to group them together or associate them. And the third Gestalt principle is similarity. When objects are similar, we also tend to group them together. So we tend to group objects together if they are close to each other or if they're really similar to each other. So here is figure and ground, right? So this is a pretty familiar optical illusion probably for most of you, right? And so depending on what you choose to focus on determines what's the figure and what's the ground, right? So the figure might be the purple piece for you, and then that is two faces, right? But if the figure becomes the white piece, then all of a sudden you have a vase or a vase, if you want to be fancy, right? So what we choose to make the figure is going to influence what we perceive. So in terms of shape here, so here is an example A of things that we use closure for, right? So even though there are gaps here, we can tell that the first thing is a triangle, the second thing is a square, right? The third thing is an animal of some kind. I don't know if it's supposed to be a zebra or what it is, honestly. But we can tell it's a animal, right? So when a person sees a disconnected or incomplete figure, they see a whole. So we see what's there, even though it's not actually closed. Proximity is that second Gestalt principle. When we see objects close together, we tend to group them together. So people looking at B tend to see 4 columns rather than 16 individual squares. Makes sense. And the third Gestalt principle, again, is similarity. When objects are similar, we tend to group them together. So I think that this is too small on your screens to be able to see this. Usually this is like blown up in the classroom. But the left one in C has vertical columns of circles and squares, and the right one has horizontal rows of circles and squares that alternate. And we will focus on that rather than alternating because it's easy. So in terms of visual perception, another important component is depth perception. And I have several slides about this that I'm not sure I can get through in 2 minutes, which is about what we have left in class. So we will just pick up with depth perception on Monday. So I will close out the PowerPoint here and stick around for a couple minutes if anyone has any questions. If not, everybody have a good weekend. Stay safe. I don't know if y'all saw the email, but there's 10 cases now on campus. So don't put yourself at any risk. Wear your masks, all that good stuff.